The overall goal of this procedure is to obtain simultaneous electrophysiological recordings of a motor neuron action potential and the associated synaptic response in target muscle. This is accomplished by first removing the skin on the top side of the fish. The second step of the procedure is to expose the spinal cord by removing all of the overlying dorsal muscle cells. The third step of the procedure is to expose the target fast muscle by removing the upper layer of slow muscle. The final step of the procedure is to current clamp the caudal primary or cap neuron and voltage clamp the target skeletal muscle. Ultimately, results can be obtained that show the synaptic responses in muscle that are associated with firing of motor neuron action potentials through simultaneous current clamp of the motor neuron and voltage clamp of the target muscle.
Larval zebrafish offer a combination of advantages that facilitate paired motor neuron skeletal muscle recording. The small size of muscle permits effective voltage clamp, and the stereotypic features of the cap motor neuron allow easy identification and access in vivo. Now, I'll step you through the procedures that are required to establish patch clamp of both neuron muscle.
Prior to beginning the procedure, prepare 5 key solutions, which include bath solution with tricaine, bath solution with formamide, neuron internal solution, and muscle internal solution. Next, collect a single larval zebrafish between the ages of 72 to 96 hours and place in bath solution with tricaine. Within 1 minute of exposure to the anesthetic, the fish will not respond to touch. Place the fish on top of a plastic dish and, using a stereo microscope, decapitate with a double-edged razor blade. Transfer the fish to a glass recording chamber coated with Silgard and filled with bath solution. Create a skin flap near the rostral end of the fish with a pin that will provide an adequate grip for a pair of fine forceps. Remove the overlying skin by gripping it near the rostral pin with a pair of fine forceps. Peel slowly in the caudal direction to remove the skin. Treat the fish with 3 milliliters of bath solution with formamide for 3 to 5 minutes to block muscle contractions that might interfere with recordings. Wash 5 times with normal bath solution. Remove part of the superficial layer of muscle over 5 to 6 segments by gently scraping with the side of a tungsten pin. Transfer the fish to an upright microscope located in a Faraday cage to shield electrical noise. The microscope is equipped with a fixed stage, long working distance 40x objective, and zoom of 0.5 to 4x. The microscope is mounted on a motorized XY translation stage in order to move the preparation between nerve and muscle under high power. The use of differential interference contrast optics helps to visualize the neurons. Under 0.5x zoom, use a wide-bore 15-micron pipette to remove overlying muscle and expose the spinal cord. Negative pressure is applied through a 3 cc disposable syringe, and muscle cells are removed one by one. This procedure is repeated for 5 to 6 dorsal segments of skeletal muscle. The same wide-bore pipette is used to also remove the upper 2 layers of ventral muscle to obtain target fast skeletal muscle. This completes the preparation for paired recordings, and the microscope zoom is increased to approximately 1.6x. The cleaned segments are scanned to locate the cap neurons. Each hemisegment of spinal cord contains one large 10-micron diameter cap motor neuron. This teardrop-shaped soma is located superficially under the spinal dura near the caudalmost end of the segmental boundary. A cap neuron is chosen for recording, and the associated caudal segment ventral target muscle field is scanned to check integrity. 2 patch electrodes are positioned for recording, one for the neuron and a second for muscle. The upper electrode has a shallow taper for penetration of the tough spinal dura and a tip opening of approximately 2 microns. The lower electrode has a steeper taper for low-resistance voltage clamp recording of skeletal muscle. Position the neuron electrode at an angle of approximately 30 degrees to penetrate the spinal dura, about 1/2 segment rostral to the selected cap neuron. Advance the electrode using an axial drive manipulator while applying a gentle continuous positive pressure of approximately 60 mm mercury. As the electrode breaks through the dura, the cap neuron may be displaced by the perfusion from the electrode, requiring a readjustment of the focus. When the electrode touches the cap neuron soma, positive pressure is released and usually results in immediate formation of a gigaohm seal. However, in some cases where a seal fails to form, it becomes necessary to apply a gentle negative pressure. After seal formation, the electrode potential is adjusted to -80 mV and application of negative pressure ruptures the cell membrane. The cap neuron is characterized by an input resistance of 140 to 180 megaohms and an action potential that overshoots plus 40 millivolts. This is tested by injecting incrementally increasing depolarizing steps under current clamp until the action potential is elicited. Next, the microscope is relocated to ventral muscle using the XY motorized translator under high power. A fast muscle cell is chosen from the target field. The muscle electrode is advanced toward the target muscle cell under positive pressure, which when released forms a gigaohm seal. The electrode potential is adjusted to minus 50 millivolts to inactivate sodium channels, and under gentle suction the cell membrane is ruptured. A drop in resistance to 100 to 200 megaohms and the sudden appearance of the large capacitive transients signals entry. To test for paired recording, the motor neuron is depolarized by incrementally larger injections of current until an action potential is elicited. At this point, an end plate current should be elicited in muscle. Continued stimulation of the motor neuron at 1 Hz results in end plate currents without failure. As the stimulus frequency is increased to 100 Hz, the end plate currents fluctuate in amplitude and undergo functional depletion within 10 seconds of stimulation. Typically, the neuron recording is stable for up to 1 hour, but the muscle cells deteriorate as reflected as an increase in the series resistance. When this occurs, the experiment is either terminated or another muscle cell is patch clamped. All recordings should be completed within 1 hour.
Once mastered, a paired recording can be completed within 1 to 1.5 hours. It's also possible to obtain 1 or 2 additional pairs in the neighboring segments. A typical day of recording will produce 2 to 3 pairs. Paired motor neuron skeletal muscle recordings provide a unique opportunity to explore the fundamental workings of neuromuscular transmission. When used in conjunction with motility mutant fish lines, it can pinpoint the source of neuromuscular dysfunction and provide new insights into the mechanisms underlying human diseases such as myasthenic syndrome.